Following on from Angus's talk earlier this year and his visit to Ireland, we wanted to build on that knowledge of the rubus species found in Ireland, Northern Ireland, with the aim to promote more detailed recording in the future. So this workshop will be one of several events over the coming year that are focusing on rubus identification. So now I'm delighted to introduce tonight's speaker, Angus Hannah. Angus is the author of the BSBI Handbook Brambles of Scotland, VCR for the Clyde Islands, that's VC 100. And he's currently working on a handbook specifically for the Brambles of Ulster. So we're absolutely thrilled to have him here this evening just to give us a quick talk about how Brambles might have arrived in Ireland, just a few words, and then we're going to move on to looking at some Bramble identification together. So Angus, thank you very much for coming to speak with us. Over to you.
Well, thank you, Kim, for those kind words about me and so on. And I should see your next screen coming up in a minute, should I? Yes. So I'm going to start by saying a few words about different thoughts of how brambles might have travelled in coming to Ulster, by what means they might have got there. And for a long time, there's been a thought that there were at one time in the past land bridges connecting Ireland in various directions to other places, such as France, Cornwall, various parts of Wales, Cumbria, south of Scotland, and Islay and other places to the north. But I think there's now a scholarly consensus that there is no such thing that's survived long enough to have any relevance for the arrival of brambles or any other similar plants that would be killed off by a long cold spell, which is exactly what Ireland suffered from in the period between about 11 and 10,000 BP, which is considerably later in time than the present opinion is that land bridges ceased to exist anywhere by about 16,000, so about 5,000 years too late for that. So that's really all I need to say on land bridges, because they're not really pertinent any longer. Bird-mediated dispersal is a much more controversial topic that has been widely promoted by bramble experts and bramble enthusiasts generally as being a major method, a major vector for the distant dispersal of brambles. And it is my considered opinion, having looked at all the evidence, that this is completely false. I don't believe that birds have any significant contribution to make or have ever made such a contribution. And there are various reasons to support my position on this. First of all, I came to it from learning a bit about the ornithological side of it. That's to say, what birds, such as thrushes, that are generally considered the most suitable ones, actually do eat. And one of the things they do not eat, by and large, are brambles. This has been proved in extensive ornithologist surveys, especially the Snow and Snow survey of the 1980s. They studied them for 5 years, and they observed carefully all the birds that ate any kind of fruits or berries that were growing in the place where they were, which was actually in England, in Hertfordshire. But it would apply equally well anywhere in Britain, I think, or in Ireland. And the result was that the only kind of thrush-like bird that they saw ever to eat them were blackbirds, and that was only for a short time in the season when there was a gap between other fruits. They're not a favoured fruit. And other ornithologists have said things like, you should make a note whenever you see a bird eating a bramble, because you won't see it very often. You won't make a long list in 10 years, as he said. And so that's one reason why I'm not going to do it very often, because they don't very often eat them. But then there's a second difficulty, which is that if they do eat them, they don't remain in the gut for very long, because it's a physiological necessity that if you're eating fruit, you have to eat rather a lot of it to make enough, basically get enough calories out of it. And so they have to dispose of the seed or stone element of the fruit very rapidly. And for this reason, birds much prefer fruits that have a single large stone in, like a cherry, rather than a bramble that has a large number of small seeds, all of which have to be separated out and sifted from the pulp that they want to digest. And the net result is that they don't keep them in the gut for any length of time at all, beyond at most about 40 minutes. So they're not going to travel a huge distance in that sort of time. So those are two explanations why it seems to be the case that birds don't very often. But I think there's a much more significant and, decisive reason for rejecting them. And that is the actual distribution of brambles themselves. This really refutes the idea, because the distribution of species brambles is of a nature that each species is in a fairly well-defined locality. And it's been there for a long time. There's no doubt about that. And Newton himself, when he was thinking about the distribution of brambles, was aware of this fact, and he came out with a very good idea about florulas, of groups of brambles that are associated in one place or another. And he pointed out that in many cases, brambles had been blocked from getting any further by such topographical features as a small range of hills, or a boggy area, or a wetland or whatever, or even a limestone plain, because most of them don't like lime. So he drew attention to these things, and he obviously saw that that was the case. But of course, if birds were capable of transporting them any distance, none of these obstacles would be any effect to them. They would easily have got to the other side of a small hill range, or a small plain or something. They're going to be able to transport them hundreds of miles, as has been suggested by some of these advocates of the theory. So I think on the whole, we have to set aside the idea that the birds have any really significant role in long distance dispersal. I think they disperse them a short distance quite often, they very probably do scatter them along the hedgerow or whatever. There's a remark by Watson, who some of you will know, grew a great many species in his garden in order to observe them. And many of these, of course, didn't come from his district at all, and were quite unfamiliar there. And he says when he walked along his lane, he would see some of these, unfamiliar ones scattered along up to a couple of 100 yards away. The interesting thing is he didn't see them any further than that. If birds were taking them any distance, they wouldn't have stopped at 200 yards, but evidently they were living within their own territory, and that was as far as they went. So that's bits of evidence that come together over the course of time, but I think we have to really look for other things than birds. So that brings us to what else could it be? Obviously, brambles didn't travel across Europe without some vectors to assist them. And ancestrally, there is a good reason to believe that they are adapted to mammals as their vector, because, as I said about the nature of the fruit already, it's not the kind of fruit that birds like. Also, it hangs low down towards the ground, which is another thing birds don't like, because it's too easy to be grabbed by a predator if you're too near the ground. You can't see what's coming. So the various reasons why you would believe that they have been adapted ancestrally, long time ago to mammals. And I believe that mammals were responsible for getting them migrated up from their refugia in places like Iberia, Italy, and Balkans up to where they now are in Central and Northern Europe. And of course, these mammals are, or were, common in those days, larger omnivorous animals like bears and wolves, and still badgers and foxes around possibly have a little role in it. But the other one that we tend not to mention is humans. And of course, humans have also been around as mammals for a very long time. And they are omnivorous, and with a particular interest in eating fruit rather more than many of the others. I'm sure they had a large role in it right from the start, but just looking at Europe in the immediately preceding the Holocene and during it, I think we can say that humans played quite a big role in the migration. And once they got into Britain or Ireland by one means or another, the means being, in the case of Ireland, very probably by boat, as they had to cross water, given that there were no land bridges. I don't think that they crossed by any other way than in boats, really. So humans were very significant at that time. But then there came to be a real change at some point in the middle period of the Holocene, which seems to be, to my mind, it seems to be coincidental really with the arrival of Neolithic ideas and people, and the settled farming lifestyle taking over from the much more mobile Mesolithic one. And this meant that people didn't move around so much, and humans were much less effective as vectors. And in addition to that, as their population increased and they farmed more intensively, as you might say, they tended to do in all the larger mammals that were also acting as vectors. The bears and the wolves and so on were driven to the margins and eventually eliminated. And so for a long time, brambles really were stranded with no vectors at all that could help them to move any distance. And that, I think, is how they came to have the kind of distribution they do, in which there's a fairly well-defined area in which each species occupies. And it's very noticeable in the last century or two, there has suddenly begun to be a great deal of movement. I want to hear me a bit louder. Is that right? Okay. Maybe I'm too far away from the microphone. I'll lean forward a bit more. Yes, it joins in really with what I was saying about birds, because we have, for instance, in the north of Scotland, several species of bramble that don't have any range further south. They just stop at a certain point halfway down Scotland. And every year, thousands of thrushes come through there and could very easily have transported them a bit further south if they were able to, and if there wasn't something preventing them. So they have never done so. So at some point they managed to get up there to the north of Scotland, and that, I presume, was when the mammals were active and people were active and took them there. And then, as I say, for quite a long time there wasn't really much in the way of vector available for them to move. But that's all changed in the last 100 or 200 years, from, as we see in Ulster, when the hedges came in, we began to plant a great many more hedges. And the quicks that were used came largely from England, I think, because they did contain the hybrid hawthorn. This is rather interesting because it doesn't occur otherwise in Ireland. It doesn't occur in Scotland. It occurs in the southern half of England. So one presumes that the cuttings came from there. And at the same time, it's very probable that brambles came in with them accidentally in among other plants. It would be very easy the way they would bundle them up in large bags and just shove them in, and either seeds in the soil that came with them or actually rooted cuttings could have been there. You don't know. But this explains why the odd brambles can crop up in hedges out in the country. And then, of course, in the domains where they were planting up larger woods, they would also import trees from various parts of Britain. Some cases maybe from the south of Ireland, but very often, more often probably from England or Scotland. And these, again, could easily bring in other species with them. And we know that this has happened frequently in recent years because there's been a huge increase in the number of species known in Ulster. when I first looked at the Bramble Atlas from 2004, there were 59 species recorded on the maps in Ulster. And now there's 123 on the list. It's more than doubled in that time, just from the forays being made by experts from England, such as Alan Newton and David Allen, coming and looking, see what's here. But they haven't, recorded widely or generally. They've just looked for these unusual plants and they found them. So that's very interesting, but there's a lot more we need to do to fill in the gaps. So shall we move on to the next part of the talk? Okay, you're asking what makes brambles hard. Well, I think the simple answer to that is that they have so many characters that you might look at. A vast number of characters that might be significant, and the problem is you don't know which ones are going to be significant in any one species until you've identified it. And this is partly why it's quite difficult to make a key, and the problems that you may encounter in the key, I apologise for, but I can only say that they do arise from the character and nature of brambles themselves.
Just before we go on, folks, I'm just having a little challenge forwarding the key on. So I will share that with you by email as well after, but we will be showing the key on the screen. So we'll be showing from couplet 8 on the screen. You just keep an eye out. If I can get it dropped into the chat, I will do that as we're talking, okay?
Okay. Well, it tells us here, very true, that stems and prickles can be as important as leaves. They are, I'm sure, just as important, sometimes more so. You never know in advance. Some species, the one is more important, sometimes the other, but stems and prickles are always important, really. And you have to know which stems, because the prickles and everything else on the stem is different according to whether you're looking at what we call a primocane, which is a this year's stem coming up from the ground, and not in the spring of the year, but in the latter half of the year. Whereas the other stems that are there, which are the ones that the flowers grow off, are last year's stems, and they have already lost a lot of the prickles and other things that would be useful identification. Because of the enduring the hardships of winter, they're no longer there. So you have to make sure you get the right stem. And if you look at a rachis stem, which is actually, at the inflorescence where the flowers are coming off, you'll find that's different again. So you've got to separate the different types of stem you've got. And it says photos may often lead to species groups and not to species names. This is perhaps true to an extent, though I hope we can get past that. Sometimes you have to settle for a group if you're doing something too difficult. Hopefully the common species can be identified without that degree of difficulty. Shall we move on? Yes, so this is a little summary of what we might be looking at on the stem. This is a specimen stem here, which has a fair mixture of things. The prickles themselves are not very numerous and not very strong, but there's lots of hairs and there's lots of acicles. I think there's quite a few stalk glands, so you'd need to study a little bit closer up to be certain of that. I'm fairly sure there are. Acicles are little needle-like prickles that just don't have anything else. There's no base to them. They just stick out straight from the stem as if somebody had pushed a needle through it. Stipules, yes, well, they're these leaves that you see coming off the stem at just off the petiole there, beyond the bud. They're not of any very great importance for identification, except that there's one group of brambles in which they're a bit broader than in the others, but I've never found that terribly helpful, really. I wouldn't suggest they were a great, primary thing to look at. The armature on the stem and rachis is, as I said, different. Yes, you have to discriminate between them. And they can be very different in some species. Okay, on to the next one. Leaves, yes, there are lots of characters in leaves. As it suggests, you might look at the shape of the tip and of the base. The tip is usually considered to be either acuminate, which they are in this one, especially the left-hand leaf there is a good acuminate one, or they can be cuspidate, in which case the tip is, it's hard to describe really, isn't it? The leaf goes rounded more and the tip comes out more suddenly from it, and we'll see an example at some point. The terminal leaflet is the one that attention tends to focus on because it's perhaps more consistent than the others. That's the one pointing straight up in this photograph, of course. And the serration is useful, but it is often variable and it's not the easiest character to use. But you can generally say whether it's fairly rough or fairly smooth, fairly coarse or fairly fine, that kind of thing. And sometimes the margin is decidedly undulate and sometimes it's quite flat. The undersurface is quite useful. The upper surface is not so helpful. The lower one is the more useful surface, but we often don't see it, especially in photographs, unless you make the effort. And another one that's not mentioned here is the character of being pedate or digitate, which is well illustrated in this case, where it's clearly digitate, where the stalks of the basal leaflets arise from the same point as the laterals. So all four, or no, five indeed, are coming off from the same point. In a pedate leaf, the two basal leaflets would be stood off the stalks of the lateral leaflets so that there was a distinct distance between them. And that's a useful character in quite a few species. Though again, you get some that are intermediate or unreliable. Here in this texture, well, that's all useful, but it does tend to vary from one time of the season to another, so you have to be careful. Okay, next one. Flowers, all right. Well, flowers are probably the most useful single thing. I would be reluctant to do an identification of sort of a species I didn't know at all without seeing a flower on it. Once you get to know them, you can manage without, but at first I would strongly recommend you stick to plants that have flowers. The flower colour and the petals are not really the most useful thing. The colour is not very reliable. Well, some species that are consistently pink and some that are consistently white, but a lot of them, you'll read the description, it says pale pink or white, and you can get something in between. It's a mixture of the two, and pink ones can fade, and sometimes they just, for some reason, don't come pink on a particular bush you would expect them to. But there's features in the shape of the petal that are very useful. The notching at the tip sometimes, or if they taper in a particular way to the base, if they stay in a cup shape or if they lie flat and such like. The shape of the whole inflorescence is sometimes helpful. It's something that tends to be seen more clearly and obviously in herbarium specimens than it is in field, because we often see rather poorly developed inflorescences that are not typical, whereas the specimen that's been collected will be the best available one, and that will be the kind that's described. So I have doubts about the usefulness of it in many cases. Sepals are very helpful, but again, there are several points you have to be careful about. You'll be told that they reflex or that they are patent or that they are ascending, erect or clasping. But these things vary at different times in the process. Sometimes the flowers reflex right away, like in this one you have here, you see them sharply reflexing. But In some species, later on, they'll rise up and clasp the fruit, whereas in others, they'll just stay reflexed all the time, and so on. And the other point about sepals is that they can be long or short-pointed. And that's useful too, but you have to know that the long points tend to occur only on the terminal flower. You have to remember lateral flowers tend to have shorter ones, and don't be confused by that, because it doesn't always tell you in the book which ones they're referring to. The filaments, well, the colour of them is fairly reliable. It tends to be white, but it's sometimes pink, especially if the petals are strongly pink. The filaments tend to be pink too. And the length of them, I suppose, which is really just the length of the anthers, you know, the whole stamen, really. And that's measured in a rather peculiar way. In comparison to the styles, you have to look at the flower sideways on to see if the stamens are long enough to hide the styles, or if you can still see a bit of them sticking out. Well, sometimes the stamens stand up straight and seem very tall, and sometimes they'll lie out flat and be just as long, but they get called short because they're not hiding the styles. And the other important thing about the stamens is that the anthers may be hairy sometimes, and that is, when it happens, is extremely helpful, because there's not many species that are like that, and once you find them, you can say, hooray, you found something, and you're almost certainly able to identify the species. I have offered a short key in the book which deals entirely with the species that have hairy anthers, and that will save you some time, but sadly for you, you don't have as many hairy anthered bushes as we have in Scotland. We only have 3 species that are common that have them, but they are all common, and that means you get a high proportion of hairy anthers, whereas I think in Ulster, none of the hairy anthered ones are particularly common. And the hairy carpels are very useful, but you need to know, which I didn't know for a while, and since I learned it, I find how helpful they are. You have to remove some stamens at this stage that you're looking at when the petals are still on the flower, and you can then see the carpels, and they will either be hairy or not. But if you wait until the petals have gone and the fruit's starting to develop, then hairs may well have fallen off. Some species will keep them right until the fruits turn colour, but not generally. Very often they fall off. So that's a quick run through most of the characters you might look at in a flower. And fruits, well, I like to see the fruits on them, but they're of relatively limited use in identification. The best thing about them is that some species, they stay red for quite a long time before they go black. And if they do that, they often have a distinctive shade of red, pinkish or an orangey or a corally sort of colour, depending on the species. And that's quite helpful. But as to the size and shape of them, I don't think it's much use, because as you can see there, the terminal one is always much bigger than the other ones, and often a different shape as well. So you have, possibly big and smaller ones on the same stem, even on the same little bit of the inflorescence. Though some species have consistently smaller fruits, this is certainly true. You'll get to know them by that, but it's not really enough to identify anything by. Okay. All right. So we're going to go through an example now. I'll leave that to Kim to talk about.
Okay, so we're going to run through a set of photographs and see if we can work it out from the taxonomic key what species it is. I will send the key on to everybody by email after the talk. I don't want to potentially ruin the screen share by going to look for it now, so I will send it afterwards, but we will put each couplet up on screen throughout our talk so that we can work it through together. We're going to start at couplet 8 though, aren't we, Angus, because the first few
Yes, the
A few
first few
steps or
are for the corallifolian section, and I haven't put in any of that
yeah,
kind for tonight, so there's no
yeah.
point in doing that.
Exactly. So we'll skip straight to number 8 on all of the ones that we're working on. But when we send it on to you later on, you will have the full key that Angus has kindly put together. So here's our first example, and we're going to start working out what it might be. So Angus, if you are able to kind of talk us through what those three options mean and how we might work out from this photograph which one is the most relevant, that would be amazing.
Yeah, very good. So here we're looking at the stem, obviously, and looking to see whether there are, this is a primocane that is in the photograph, so that's what we're referring to. We want to see whether there are stalk glands, acicles, or pricklets on it or not. That's the first part of the question. The second option is that there are acicles, short glands, pricklets, or prickle stumps, usually present in varying quantity on the stem faces. And the main prickles are all on the angles of the stem, which of course would be true in the first question as well. It would be on the angles principally. Or the third option might be that prickles are found all around the stem, with longer ones on the angles but shorter ones elsewhere, and so on, lots of pricklets and acicles. And I think it's fairly obvious to you which it's going to be.
So from that, we just want to make sure that we're all looking at the same things here. Are we saying that there are stalk glands, acicles, and pricklets are more or less absent from primocanes? That they are present in varying quantities, or that they are all around the stem, unequal, and longest on the angles? So, which one would we be saying here? And so I'll skip on to the next one, and we're saying they're more or less absent from the primocane. Is that right, Angus?
Yes, that's right? That's what people are saying. Good.
Yeah, but this is our example. We'll just walk through, and then on the next example, we'll get people to take a vote. But for this species, we'll work it through for them. So for this one, then that is saying that we're going to go on to number 9. So we'll skip on to the next section.
Right, so now we're going to look at the prickles again in a little more detail. Are they mostly straight, with some turning slightly upwards towards the tip? Or are they down curving, especially on Arachis batis, in anywhere really? Are they down curving or a bit hooked? Or are they mostly straight, whether they might be, let's just say straight out, or they might be declining, but that's the real distinction. The straight ones, or the curved ones, or the curved up ones.
And so for this example, we would say they were down curving. Is that right?
Yes, I think you probably would have no difficulty in saying these are down curving.
Okay. So then we'll move on to number 11. So we have 2 choices on this one then.
Oh. Well, this asks whether the leaflets are deeply incised, the terminal usually being dissected into 3 to 5 deeply toothed segments, or not. Basically, I don't need to go through all the details of the other plant.
So we're happy enough then that if it's not deeply incised, that it must be couplet
Yeah,
B, which
yeah,
puts us to
that's right. There's no need for me to have put all that in the key, really. I should take that out.
all the work in progress. So we're saying that example 1 is Rubus numeralis. Okay.
Yeah.
So that's our first one. So now it's your turn, everybody. And what we will do is I will put up a wee poll, and do bear with me. I very much hope this works. I have a wee poll. Can you all see it? Has that popped up? Okay, so this is our next species here. And what we'll do is for each example, we have a few more species to go through, but for each couplet, I'll put this poll up and you can say A, B, or if there is C, C. And we'll just see what everybody thinks before we actually give the answer, okay? So let's go on to our first one, example 2. And we're again starting at number 8. We're skipping the first section of the full key. So we're looking at stalk glands. Sorry, go ahead, Angus.
Does everyone have the poll thing up on the screen? Because it's rather blocking
Oh,
the.
you should be able to move it. Are you able to move?
Should we move it about? Yeah. No. Is it here? Yeah, so
I can't see how it's appearing for everybody else, so hopefully you
no,
can move it
yeah,
out of the
no,
way if it's in the way.
yeah, move it to the far corner, I suppose.
Mm,
It's not so obvious. Anyway, okay. If it's in your road, just minimize it. You can
yeah.
presumably do that.
Okay, so there's just a wee question there that I don't think I understand number 8. So um, what we're saying there, right, is I guess that the first kind of set of couplets from number 1 to number 7, we've pre-answered those before the workshop because all of the species we're showing are starting from this point on the key. So rather than going through the same few questions over and over and getting the same answer, we're skipping ahead here. And Angus, you might just explain what that sentence means, that the Coralifolian characters are not present. So what
Well,
are
yeah,
those
so
characters?
Coralifolian characters are just a group of characters that enable you to see that a particular species is in that group. It's be things like well as any one of them might crop up in the other lot, but it's a collection of things, including the leaves that are very conspicuously imbricate very often. And there is a question about the stipules that I mentioned already, that they are broader. If you have any stipules and can measure them, they're usually more than 2 millimeters broad. And the flowers just look different. It's a thing you just get to know after a bit. It's very hard to define them actually, because any one definition that you give, you'll be able to find a counterexample to it. But if you take the group as a whole, you get to know it, and you just have to accept really that we're not doing that group tonight. But well, you won't need to worry about it. At some point, they're not very common in most of Ulster. The only one that seems to be a bit common is latifolius, which is common on the coast of County Down. And the other one that's a bit common is nemorosus, which is in the southern parts of Fermanagh. But otherwise, you're not likely to meet them very often.
Okay, so just in the chat, we said we chose that pricklets were not present last time, and they do seem quite big and obvious. So if you could give us a little bit of a steer on not necessarily the answer, but what we might be looking for here. Is
Maybe
is it possible
pricklets.
that we'd be starting off on the same path as last time?
Yes, it's
Or
an important distinction, of course, between prickles and pricklets. These things you see here on this stem are prickles, and so were the ones you saw on the other one. But a pricklet is a very small prickle, about, well, certainly less than half the size of a big prickle. And it would only be, the word would only be used if there were big prickles as well. So you'd see big ones and then you would see very small ones. Often they're only about 2 millimeters, but they can be as much as 3 or 4 in some circumstances. But they're definitely less than half the size of the main prickles. So what we're saying is absent are any small prickles. We've got nothing but ordinary big ones.
okay, okay. So if you've got your poll open and you want to have a vote, if you don't want to answer, that's no problem. We'll give it another few seconds and then we'll look to see which one most people have gone for. Still votes coming in. This is great. Okay, and we're going to close it now, and hopefully this shows you. Can everybody see the result there of the poll? So most people selected A, so let's take a look. And yes, we went for A again there. So that's going to move us on to number 9 in the taxonomic key. So let's move over to number 9. I'm just going to stop sharing that poll and see if I can relaunch it for this one.
So this is, again, a question that looks at the prickles in some detail. It wants to know again, as it did last time, indeed, whether they're basically straight and sticking out, whether they're hooked and down curving, or whether they're tending to curve up the ways.
And it's helpful to get an idea, if we can, of which direction this bramble actually is pointing, which way is up and which way is down. Is this right
Yes, it
way
is
up or?
something you have to think about. And in this case, the bramble is sort of halfway upside down, I think, because I think the
Mm.
the bottom right of the picture will be the top end.
And can we tell this from looking at the
Usually,
stipules and the leaflet in the
I can't
top left
see
corner?
it very well because of this, but oh yes, you see, if I move my pole thing out the road, I can. Oh no, made it big now. Minimize it. Yeah, that's better. I don't need to see it really. Yes, you can tell that's certain because where that petiol's coming off there, that's a leaf, you see, but there's a bud in the axis of it there, and that's always above the petiole.
Mm-hmm.
So that's definitely the case that the top end of the stem is at the bottom right corner of the picture.
Okay, folks. So what do we think then? Are we looking at A, B, or C?
Remember that we're not looking at the prickles on the petiole, it's only the ones on the stem. You can see there that they're somewhat different on the petiole.
And this one is a little bit more challenging this time because the bramble does appear to be slightly down. Okay, is everybody finished? We'll give it another few seconds. Okay, here we go. And I'll share that result there. So it's very, very mixed on this one. So the most people actually said C, prickles mostly straight, patent, or declining, or a few slightly curved down. Quite a few
I
people
think that
did
would be
say
the
B.
correct answer. C.
Okay,
I'm glad a majority thought that, but
yeah. But it is a challenge because I suppose it's quite close to B as well. So here we go.
I know it's a difficult question. People will always have problem with this question, but I can't think of a better one that will get you into the next stage. You know, it's these early stages of the key that are really difficult.
But it's very handy to talk through it together as the group. So here we go on to number 12 then, and we're looking at stem prickles. The stem hairs on this occasion, sorry.
Stem hairs, yes. So is the stem densely hairy, moderately hairy, or is it glabrous or just very slightly hairy?
And just to explain glabrous, just in case there's anybody on the call who wouldn't be familiar with the term.
Well, glabrous is just a term for the absence of hairs, really.
Thank you.
And we're not talking about the petiole, remember, so don't
Mm-hmm.
worry about the
Just
fact. You can obviously see some hairs on the petiole there.
so for the main stem. So if everyone
Yeah.
gets their votes in, and then we'll see. Okay, I'll end it now in 3, 2, 1. Let's share that. And I think that's fairly conclusive. Everybody agrees with number C, or letter C rather. And so we'll move on to number 17 now. Okay, let you talk through that.
This shouldn't be too difficult either, really. A lot of leaves have white felting beneath them, and some species, and a lot of species they don't have. It's useful. Sorry, this thing keeps jumping up, getting big.
Sorry, it's because I keep resetting it after each question. Okay, so we've got leaflets. Are they pale or white felted underneath, or are they green? And I think most people are going for one particular answer here. So we'll end that now and share the results. So pretty much everybody went for A. It can be, I suppose, difficult if you don't have the context of what might be green to compare it to.
Well, you see the upper side, it's obviously not the same color as the upper side.
Okay, so we're going to move on to number 18 now. Here we go, and I'll just reset the poll. So leaflets chalky white felted beneath and petals pink, or leaflets pale or whitish beneath and petals
Yes,
pink
this is
or
a bit
white.
harder because you haven't got the experience to know what's meant by it, really. That's why it's difficult.
So how would you tell chalky white felted beneath? What would that look or feel?
Yes, it's a difficult thing to say for certain, but you get to know, the range of colour that you have. You have quite a lot of species in which the leaf is, as you might say, greyish white or greenish white. And there's no pure white, but some are definitely more obviously white than others, and this is one of the obviously white, really.
Okay.
I don't think it's fair to ask you to judge if you haven't got a means of actually comparing it to other species. It's obvious that it's very different from the upper side of the leaf, but you don't know that there might not be other species that have it similarly to that, and that would call just a bit whitish or pale or whatever. It's just difficult. But that's about as white as you get. I think I have to tell you that, really.
And just to look at the petals then, could you talk us through what we might be looking at there on the petals? Like how would you decide whether they are fully pink or white? Does that mean some might be pink and some might be white, or does it mean pink in part of it and
No, it
white in the
just
center?
means that some bushes might belong to a species that has pink petals and some might belong to a species that have white petals, because that's the other answers, There'd be several species involved if you went down that line.
Great. Okay, I think
This
everybody
one,
stopped. Oh, go
this
ahead.
one always has pink.
Okay.
Yes, I think people need a bit of help at that point,
Yeah,
really,
it is a
because
tough one.
there's no way you could actually know the difference until you have a bit more experience.
Mm. Okay, so I think if everybody's finished voting, let's share that result there. And remember, folks, the Q&A and also the chat functions there. So if you have any questions or comments that you want, we can answer those as we go along. So it was very split here, but slightly more on the A side. So what do you think, Angus? Would you agree with A?
Um, I think so. I can't
A.
remember which one was A now.
It's leafy chalky white felted beneath with petals pink, or
Oh
B was
yeah.
leaflets pale or whitish beneath and petals pink or white.
Yes, yes. I think that's the one I was hoping for.
Okay, so A
And
is
I learn a lot about doing keys with this. It's very helpful.
yes. So there we go. So we're saying it is A, and that takes us to number
And I
19. So I'll
think
skip on and
anyone
I will
should be forgiven for having got that one wrong, though I don't think it.
absolutely. But it's good to work through it together, and it's interesting to see how divided the split was on that.
Mm.
So let's have a look now. We've got two options on number 19 here. Bushes in all parts large, stamens very long, mainly near habitations, spreading cultivar, or prickles often triangular, strong, and the stamens are short, level with styles.
So the main difference here is that we've got stamens, and we've got stamens very long in the one case and stamens short in the other case. And that's what's going to be easy to tell.
Hmm. Okay, so what
Remember,
do
I
we think,
told you that
folks?
long stamens are longer than the styles, and short stamens are shorter than the styles. They're about the same length as them. So
Okay, so it looks like most people are going for the same option.
I think, yes, I think most people would get that one. It's a
Mhm.
nice clear cut answer.
Great. So I'm going
Indeed,
to share the result there. And yes,
that's the
so
first time we got 100%. I like
so
to see
well done, everybody.
a good answer. That's good.
Absolutely. So we have Rubus arminius, and that
Unfortunately, you just don't always get, a nice choice like that you can offer.
yes. Okay, so well done, everybody. So we're going to go on to our next
What
example.
Almanacus, I should maybe say just a word or two about it.
Oh, sure. Sorry.
You might as
Let's go
well
back.
know it's what they call the Himalayan giant. It's a very large and rather horrible bramble, which is an invasive thing, and once it gets a hold in a place, it can spread and take over. It's still rare in Ulster, but I think it won't stay that way very long. It's spread hugely in many towns and cities across Britain,
Right.
mainly urban, but it's not restricted to urban sites.
Must be the one in your garden. Yes, I think I have some of this in
Could
mine
well
as well.
be, could well be.
We'll have to go out and ID it tomorrow then.
I think we found it in John Faulkner's garden.
Very likely. Okay, well, we move on to the next one, everyone.
Yes, let's go.
Okay, we have about half an hour left of this evening.
I should get
So we
through
want to
these.
try and get through these.
Yeah, it's just a
So,
couple.
so here's the next example, and you can see straight away that one's quite different, isn't it? So let me get the poll up again. Sorry for constantly popping this up and down. Okay, so here we are. Again, we're starting at number 8, just to skip that first section. So we're back to that very first question that we looked at. Are there stalk glands, acicles, and pricklets more or less absent from the primocanes? Are they usually present in varying quantity? And the main prickles are all on angles of the stem, or prickles all the way around the stem, unequal, longest on angles, and mixed with pricklets and acicles. So, folks, what do we think?
Course, the bit about being usually present shows that the key wasn't written for doing photographs. It was written for looking
Mm,
at bushes in the field, and you could look from one to another and see if they were all the same or not. You can't do that with a photograph. So you
yes,
have to ignore that.
it is a unique challenge doing something like this online, but I think it's a good starting point, and it hopefully it also helps people understand a little bit how important photographs would be for if you wanted to send something over to Angus. You'll get an idea now of what kind of photographs are really important and what features would need to be shown so that Angus can help you um,
Yes,
identify
this is
something.
very true, and I'm very happy always to get photographs.
Mm. Okay, so is everybody who has an idea added it in? We'll give it another few seconds and then we'll end the poll.
Probably better to look at particularly whether the prickles are all round the stem on if they're on the angles. That's the long prickles we're talking about
Mm,
when we say prickles.
okay. And so on this picture, the prickles are fairly obvious, but would the pricklets be about halfway up that
Yeah,
stem on the other side, on the left-hand side? Would that count as a
there's one
pricklet?
or two little pricklets on the left-hand side there, particularly, yes.
Okay, great. Okay, everyone, we'll close that poll now and I'll share the results. And so most people went for B. A few did go for C, though. So let's talk that through.
Hmm.
Um, what do you think, Angus?
Let me just take this off again.
Oh,
Right.
yes, sorry.
Yeah. Well, this is the question, are the prickles all round the stem? And it seems to me fairly plain that they're on the angles. The angles are not very sharp, but there's an angle running down more or less in the middle of the picture, just slightly to the left of the center of the stem, as you see.
So when you say angle, does that mean that the stem itself kind of has
Yeah,
a
yeah.
fold or a
It
rigid
has
edge?
angles and flat
Mhm,
faces. You see,
mhm.
there's a flat
Yeah,
face towards the right, and there's a flat face much more steeply towards the left.
yeah.
And the prickle on the left-hand side towards the top is at the far end of that flat face,
Mhm.
and there's one at the same position near the top that's coming straight out towards us, so it doesn't look like a long prickle, but that's on this angle. It's coming straight down, the same angle that the bottom prickle in the middle is on. And then the other four prickles on the right-hand side are all on the angle of at the end of that other face.
So
So
again, probably something that you'd easily see if you were in the field looking at it, because you'd be
yeah,
able to feel that folding or that
there's
curve in the
no long prickle on the face. That's
stem.
the important thing. You could run your finger right down the face of the thing, the flat face, without encountering a prickle.
Okay, so most people went for B, and then actually quite a few people did go for C though. So let's have a look. And it was actually B in this one. So what would be the reason that it wouldn't be C then? Would it be, is there something that makes it very obviously not C, like the prickle length, that they're all pretty much the same length? Or
They're all round the stem, you see, in C. So that
Mhm.
and here they're obviously not all round the stem because they're on the
That's the main.
angles.
Great.
The important thing.
Okay, so
In
let's,
both cases, there's plenty of pricklets and stuff on the faces, so there's no distinction in that respect. The only real
yeah,
difference is whether or not they're on the angles, whether or not the big
sure.
prickles are on the angles.
So we'll move on then to number 26 in the key. So we jumped ahead quite a bit and we're looking at this next one. And our choices are, so the stems are glabrous or nearly so, and glandular hairs are often present, or stems with variable, oops, typo there, glandular hairs may be more or less glabrous later or in shade.
That's not helpful because you don't know whether the picture was taken later or if it was taken from a plant in the shade. Except you do know really by the colour of the stem that it's not a shaded plant, because they only colour up if they get some sun.
Okay, that's useful to know.
Hmm.
So, folks, get your votes in. What do you think? A or B here?
Should tell you perhaps that hairs are always seen most easily when you're looking against a dark background at the edge of the stem, as it were, rather than on trying to look on the face of it, because they'll disappear on the face and you won't be able to see them. So
Okay.
look along the edges and see if there is a evidence of eglandular hairs, that's to say,
Mhm.
as you say, ones that are not don't have a gland on the tip. I think,
I think we have everybody in.
otherwise you have
Say
to say that it's glabrous and it's not. Well,
again.
doesn't
Okay.
look so it is.
So I think pretty much everyone stopped voting now. So we'll end the poll there and share the result on. And most people went for B.
Be
So that would bring us to number 32. And that is what we thought. Great. So let me just reset the poll. Here's our next one if you want to go through that, Angus.
well, prickles numerous are not especially numerous.
And so that specifically prickles, not anything else.
Well, again, it takes a bit of experience perhaps to know what we mean by numerous and not especially numerous, but on the whole, it seems to me that there are significant gaps that you would not expect. I think if they were numerous, they would be like they are at the bottom right there, all the way up the left as well. So it's
Great.
it's partly for me to explain what these things mean as much as for you to guess the right answer.
Mm-hmm.
So
But I think that's very helpful because most people now, I think pretty much everybody's voted. So I'm going to share the results there. And with your explanation, yeah, most people have gone for B. And that is what we were hoping to see. Okay.
yeah, they're not especially numerous. That's right, of course.
Okay. So let's go on to number 34. So now we've got our choice of three. Um,
Oh yes.
if you want to go through
No, this
anything
is
you'd
going to
like
be
to
tricky
talk.
because we've most of the things that are spoken about there aren't here because you haven't got flowers or anything to look at. You've still only got. Can we bring up the picture we started with that had that had flowers and things in it, just to let people see that.
Yes, so this one I'm going to go through a few different pictures. So
Oh, yes, you can go
we've
through.
got the
Okay.
stem,
Well, the
because
STEM. Let's look at the STEM questions first then.
just so that they're big enough that people can see it on the screen. So we'll look at the stem first.
So, since this, I've actually changed the key. I'm taking out a lot of the stuff in here. There's far more than there needs to be in it, which is confusing.
Well,
Before you
tell me if
send it around by email, I'll send you the revised key, which is
brilliant.
a bit different. Anyway,
So would you like to start by looking at the flower? We can skip backwards and forwards.
we're all right with this, I think. Prickles on the angles only, we've already decided on.
Mm-hmm.
So that rules out number C, doesn't it? Because that has them not confined to the angles.
Mm-hmm.
So it's going to be between A and B.
So I'll go
We'll
on to
go
look
on
at
to
the,
the flower then. Let's see
yep.
that. Right. And we know from what we've done before that the flower has long, do we know? We don't, because we didn't do this one, but it does, you can see from there that it has long stamens. And you can also, if you look carefully at the top of the stamens, see something else that's very interesting.
Mm. So has anybody seen anything else on the stamens that's of interest that we might have mentioned earlier on? Okay.
Anybody?
Look at those hairs, yes.
Yeah, well done, well done. Right, so we are told that Dreher Eye, the bottom one, and number C has that, but we've already ruled that out because it had tells you the prickles are not confined to the angles, and they were, so that
Mm-hmm.
doesn't matter. We've ruled out C already, and B has anthers hairy,
Mm-hmm.
and A doesn't. That should be stated, and it isn't, but I can take it that it's the case.
Sure. So if everybody has finished voting, I'll give you another couple of seconds because I see there's a few more votes coming in. So, lovely stuff. Right, we'll end that now. And most people went for B.
Yes, that's good. Yes, you
Yes.
did also have the long
Oh,
stamen,
sorry.
which were said to be short
Yes.
in the other ones, so that
And
gave
here's a
you
wee
two
picture.
reasons to go for it.
Yeah. Okay. So let's go on. There's the underside of the leaf, just in case you hadn't worked it out from the first two photos. And so there we go. So well
See,
done, everybody.
yeah, yeah. So it's quite a green underside, isn't it? Good. Okay.
Excuse me. Great. Okay. Are we ready for our fourth and final
It's
test
a
to
nice
see how we get
cuspidate
on?
tip on the leaf, by the way. There.
Oh, this one here. This is the
Yeah, and there. It's well, it's almost mucinate. You know, the name mucornulatus refers to that mucinate tip, but it's just a very cuspidate tip, really. It's not technically mucinate because there is a lamina right up to the end.
and Angus, would you just have a wee look in the chat there at the question? Uh,
Oh, can I click on chat?
yes, you should be able to see it popping up.
Oh,
And
yes, that's it.
there we go.
Now we're at the top of the bottom.
Oh, scroll down to the bottom there, just
Oh,
Finley's
yes.
question.
This is Finlay, yes. Is GI also keyed out elsewhere? Oh, yes, yes, it will be keyed out elsewhere, I think. Prickles are not confined to the angles in that species. It certainly should be. The key is far from perfect. I'll have a bit more revision to it, and in a day or two, I'll send you a better version of it. I was working on it
Mm-hmm.
this morning, actually. Yes, I've done a bit to it here and there, but there's more to do.
Yes. And I think these sessions are useful now because you'll also see how we interpret what you've written in the key as well. So that's quite
Mm.
handy having something like this. Okay. So let's go on to number 4 then. And here's the photos. And again, it's a little bit different. We're looking at something very different. Excuse me, I'm just going to cough.
Just minimize that for a minute. Can I? No. No. What can I put it?
Okay, and I'm going to relaunch the poll here so that we can skip on. And again, we're starting at number 8 because we know we've pre-answered 1 to 7 for you, and or the parts of the key that came before that. So we're starting at number 8. So we're back to that very first question we asked.
Right. Mhm.
You'll be very familiar with this question, but maybe we'll get a different answer here. So
Well,
let's get
I
some
don't
votes
think
in.
anybody's going to want number one.
It's
You've got
very
lots of
different to
stuff
the
there,
first
haven't
couple.
you? So the question is between the B and C, really, which again is, are the prickles all round the stem, or are they on the angles of the stem? Are they all on the angles of the stem, or are there some that are not? This is what it
Mm.
amounts to, really.
And we're talking about the prickles specifically all around the stem or on the angles as opposed to pricklets, is it?
Well, yes, but the other thing about this is that in the question C there, you're thinking that the the prickles are unequal and they will actually grade down to the pricklets. There'll be everything in between. That's an important thing
Mm.
which you need to understand. This series that we're looking at here, one of the defining characters is that the prickles exist as big prickles and as middling sized prickles and eventually as pricklets, and you get everything all the way down. Whereas in the B category, there's a big jump. You get prickles and then you jump down to pricklets that might be anywhere. But here, anything might be anywhere, but especially the sizes grade down.
And Harry has a good question there. Is there a good way to distinguish between icicles and hairs?
Well, icicles are stiff and will always be rigidly straight, whereas hairs are soft on the whole, or even if they're stiff, they're not as stiff as icicles. In
So
a
it might be
sense,
challenging from a photograph, but
it can be challenging on occasion, yes. If they're short, stiff hairs and short icicles would be almost impossible to
mm,
distinguish in a photograph.
okay. Has everybody who's going to vote voted? We'll end it now. Oh, a few more. That's great. Okay, here we go. And let's see what everybody thought. So pretty much everybody went for C, but it is a toughie.
I did somewhat point you in that direction. Good, I'm glad you
Okay.
picked it.
And it is C on this occasion. So we're going to go over to number 36 on the key then. And this one is either stems hairy or glabrous or subglabrous. So let me just reset that.
Yeah, so this turns in a sense on that question that's just asked in the chat there, because if they were all icicles, we would say that the stem was glabrous, whereas if there are some of them are hairs, then we would say it was hairy. You've got to try and judge whether that's the case. The icicles tend to be longer and they do tend to be very straight and stiff. If they look as though you could sort of smooth them down with a gentle touch, then there would be hairs, whereas an icicle would jag you if you tried to bend
Right.
it down.
Okay.
But there are both there as far as I could say.
So another few seconds and then we'll close the vote. Okay. So let's see what everyone said. And most people have gone for A. And that is what we are
Yes,
aiming
yes,
for.
I think we definitely have some hairs there, quite a few hairs actually.
Mm-hmm.
Quite a lot. Mhm.
So that we're going to move on to 37 then, and this is where we can decide which species. So do you want to talk us through the 3 different options maybe?
Yes, I shall try and do that.
Or is there anything in particular on this
We've looked
picture
at
of
the
it?
stem already, and we're looking
Mm,
at the prickles there. They ask if they're both straight and curved. And
okay.
the next one, prickles often abundant grading to pricklets. Well, that's true again, so I can't rule that one out.
Mm-hmm.
Stalk glands short and sparse, pricklets numerous. Doesn't say anything about the prickles in the bottom one, so we can't
Mhm.
rule anything out at the minute. So let's go on from the stem to another
Another characteristic. So here's the leaf.
leaf. All right, well, what have we got about the leaf? The first one says the leaflets are glabrous above, more or less plicate, often lobate. Of course, often is something we don't have the chance to test out here in the photograph, so
Mm-hmm.
we don't know one way or the other. The terminal is said to be broadly obovate, with an acuminate tip, often twisted.
So that's the tip is curving off to one side or actually
Yeah,
twisting around. So the bottom of it.
I think curving towards one side is all it
Mhm.
really means. So sometimes
Okay,
it might involve a bit of a twist as well.
sure.
It's more often growing towards irregularly towards one side, I think.
Okay.
Okay,
And then
that's the
for any of
first
the other options.
suggestion. What have we got in the other ones? Leaflets contiguous or slightly imbricate, and number 2, yellowish green and concave. They appear to be on the whole flat.
Hmm.
It's a slight concavity in this terminal leaflet on the nearest leaf, but some of the others are curving the other way, and most of them seem to be flat. I'm a bit doubtful about that one. And the
And then
third
option
one,
C,
what have we got there? They're thin,
leaflets.
shiny, pale green. Well, shiny, pale green, broadly ovate to elliptic. Do we know? Do all of us know the difference between ovate and obovate? Are we quite happy with that?
Might be worth just calling it out because hopefully,
Yeah, well, it
we'll
just
get some new people watching
depends
the
really
recording as well.
where the broadest part of the leaf is. If it's broadest towards the tip, then it's obovate. But if it's broadest towards the base, it's ovate.
Great. And then I'm just going to skip on to see, do we have another picture? So we
Yeah.
should have the
What else?
underside.
Ah, the underside, yes. Well, that shows quite plainly that they're not contiguous or imbricate. There's a gap between all those leaflets there. So
Okay.
that's
So,
tending to rule out the middle option.
okay. And then I'll hopefully
And
not
what
give
about
away the
the,
answer here.
is there anything about the colour of the underleaf? It is.
Um,
No.
don't think so. We have a good bit on the
Margin
flowers.
is not undulate. Yes, let's go
Mhm.
into the flower. We've got a few things here. The petals are obviously quite narrow. The sepals, you can see the sepals are very sharply reflexed, both there and there. And even up there, they're quite reflexed too. And that one's going a bit red at the top.
And then in option C, I see it also kind of mentions the sepals where it's saying they're painted or erect and then loosely reflects. So
Mm.
it's not necessarily what we're seeing here.
Reflects reddening later in the sun. The bottom one's loosely reflexed and red base later, so it's not ruling out that one. The middle one seems less good.
Mm.
Anyway,
So when we're talking about red based later, I just want to look at the inflorescence up in the top left corner of that picture where there is a kind of reddish tinge. Is that where you mean that it would be reddening, or is
yes,
that
yes, that's
okay?
right. That's what it would be.
Great? Just so people
Reddening. So it's
understand what it would.
reddening later there and also in both the first and the third example
Okay.
here.
Okay. So
We haven't got enough evidence yet to make a decision really, except by guessing, have we?
mm,
Um, it says no, we're ruling out 2, I think, but between 1 and 3, we need to try and get a decision. I'll go back to those leaves a minute. I think they were quite good. That, yeah,
so the
that's it. That's fine.
underside of them. Yep.
See, it says here that for the third option, the main teeth are prominent. And there's no evidence in here that the main teeth are prominent because all the teeth seem to be very much equal, really. There's a slight tendency for it there, but not much. So it's very much even serration. I don't like that so much.
Okay.
And we've got the acuminate often twisted tip there. The undulate margin in this, there's no undulate margin really. It's quite a smooth, flat margin.
Okay, so there's quite a few people have put their choice in. Does anybody
Okay.
else want to cast a vote? And let's see what people think. A few more seconds
Groups
there.
do look more ovovate to answer that
Mm.
question in the chat.
Oh, thank you.
It's
So if I
slightly
flick back to the picture before, and we're just looking at that tip again. Okay, I think everybody who's been voting has pretty much cast their vote there. So will I end the poll and let's see what people think?
Mhm.
Okay, and so most people went
And
with
very
A.
strong vote for A, yes.
Okay,
Good.
well done. Okay,
Well, you'll show us the answer then.
yes.
Yeah,
So rubus aciculus.
that's it, yeah.
So prickles both straight and curved, leaflets glabrous above,
Good.
sepals reflexed. Well done, everybody. That was, well, I really enjoyed that. I hope you all did as well.
I think
So
it's demonstrated to you to some degree. It's a bit tricky quite often.
it is a bit
But,
tricky, but it was for me
but
quite useful having being able to see how people are able to read the picture. And especially at this time of year when we're kind of limited going out and looking at things.
yes, that's
But
right.
yeah, does anybody have any questions? We have 7 or 8 minutes before our time is up, so we've got loads of time for if anybody has any questions, anything else to do with Rubus you'd like to pick Angus's brain on. Um, I'm going to just pop up on the screen as well some further resources, and I'll put these in the email. There'll be an email straight after this event, probably just with the feedback form. But what we'll do is I will send around Angus's queue, and he's just made those few tweaks, and I'll send you this slide here with the extra resources in as well. But if anybody has any questions or anything that you want to kind of chat through, let us know. You may just want to get back to your late dinners or whatever. Okay, no questions at the moment. Okay, well, we wrap up then. So thank you so much for that. Just before we finish, I just wanted to thank our funders, DERA, again for making the talk possible this evening, providing us with the funding for the entire project. And of course, a huge thank you to Angus for your time and the expertise and for developing that key for us for using here in Ulster, which is fantastic. Uh, Thanks as well to all of you guys for taking part. That was fantastic. I really enjoyed working through those examples with you. Let us know your feedback. Let us know if this format worked, if there's anything else you'd like to do, if you'd like to do more on this series with different brambles, if there's any other plants you'd like to do something similar in as well. That would be really good to know. And so, yes, thank you so much. And hopefully we'll see you again soon at either an in-person event or a webinar or an online workshop like this. Angus, thank you.
Okay, well, that's
Okay.
good. Let me thank everyone too, because it's really great to know there's that much interest and so on.
Yeah, brilliant. Good night, everybody, and we'll see you soon. Okay. Take care. Bye bye.